Good wishes from Dr. J. Albinovins, Assistant Professor of Botany, Holy Cross College, Autonomous, Nagarkoil. Today, let me share some insights about the thallus structure of Marchantia. Marchantia is one of the most commonly encountered liverwort. It has a wide distribution that is from Arctic to tropics and is relatively large and with a distinctive form. It is often encountered in seeps near steep rocky outcrops next to brooks and streams. Marchantia is in the phylum Hepatophyta in the plant kingdom, with division Bryophyta, class Hepaticopsida, order Marchantiales, family Marchantiaceae, and genus Marchantia. Coming into the thallus structure, the plant body is a haploid gametophyte. The gametophyte constitutes the dominant phase in the life cycle. The mature plant attains the length of about 2 to 10 centimeters. The plant body consists of a thallus, rhizoids, and scales. The thallus is fleshy and dark green in color. The thallus of Marchantia is prostrate, dichotomously branched, and dorsiventral. The dorsal surface of the thallus is divided into fine rhomboidal marking called areole. Each areola has a central pore. A single air chamber lies below the areola. The thallus bears a conspicuous midrib on the dorsal surface. The midrib is visible on the dorsal surface by the presence of a groove. The midrib ends in a notch at the apex of the thallus in which the growing point is situated. The margin of the thallus is slightly wavy. Small cup-like structures called gemmacups arise from the midrib region. The gemmacups bear asexual reproductive bodies known as gemmae. The gemmae give rise to new gametophytes. Numerous scales and rhizoids arise from the ventral surface of the thallus. The scales are multicellular, flat, purple plate like structures. The scales are produced on either side of the midrib in two or more rows. The scales are of two types, namely simple scales and appendiculate scales. The scales will protect the growing points from desiccation and also mechanical injury. The scales also serve to retain moisture below the thallus. The rhizoids are unicellular hair-like structures. They are of two kinds. One is simple or smooth-walled rhizoids and the other is tuberculated or pegged rhizoids. The smooth-walled rhizoids are thin-walled and colorless. They are produced in the midrib region. These rhizoids serve as organs for attachment and absorption of minerals and water from the soil. The tuberculated rhizoids arise from the ventral surface near the scales. Their cell wall is thick and many peg-like ingrowths called tubercles develops from the inner walls. The rhizoids grow into the soil and act as capillary conducting system. In Marchantia, some thalli produce erect, stalked upright branches at the growing apices. These branches are called gametophores. They bear the male and female sex organs. The gametophore that bears the male sex organ Antheridia is known as antheriophore. The gametophore that bears the female sex organ, that is archegonia, is known as archegoniophore. Marchantia is heterothallic. There are separate male and female plants. Antheridiopores are produced on male gametophyte and archegoniophores are produced on female gametophyte. Coming into the internal structure, the vertical section of the thallus shows two distinct regions. One is dorsal photosynthetic region and another one is ventral storage region. First, let's see about photosynthetic region. The uppermost layer of the photosynthetic region is known as upper epidermis. It is single layered. It is made up of compactly arranged thin walled cells. These cells are green in color. The upper epidermis is interrupted here and there by many openings known as air pores. The air pores are barrel shaped. These pores are analogous to stomata. Each air pore is surrounded by four rows of epidermal cells arranged in the form of rings of 4 to 8 tiers, one above the other. Below the epidermis is a horizontal layer of many air chambers. These air chambers communicate with the atmosphere with the help of air pores. The air chambers are separated from each other by means of partitions which connect the upper epidermis with inner cells. The partitions are 3 or 4 cells in height. The cells of upper epidermis and partitions contain numerous ovoid chloroplasts. From the floor of each air chamber arise assimilatory filaments or photosynthetic filaments. The filaments are branched, dwarf, cactus-like and chlorenchymatous, and these filaments fill up the air chambers. All the cells of the floor, partition wall, and photosynthetic filaments contain ovoid chloroplast. The chambered region constitutes the ventilated photosynthetic tissue. It is the principal center of photosynthesis in the thallus. The upper epidermis checks rapid evaporation of water, and the air pores will regulate the moisture status in the thallus. So the upper epidermis is protective in function. Assimilatory filaments are photosynthetic in function. Next is storage region. Just below the photosynthetic region lies the ventral storage region of the thallus. It is composed